Hello, this is Dr. Dan Guerra from VerEvMed Studios in the Pacific Northwest, United States. This is March 12th, and we're into a new video lecture series. I covered in the last couple of weeks cannabinoids and ethanol as two drugs that I wanted to discuss at the molecular, biochemical, pathophysiological level. And I hinted that I was going to do at least one more, and the third one, of course, is going to be on the opioids. So we're going to get started right now with that. And I want to say at the beginning that we're going to have a two-part opioid lecture. The first part will be mostly on pain reception, that's known as nociception, and how opioids, endogenous opioids called endorphins and enkephalins, play a role in truncating the pain response in normal physiology. And we're going to get into the drugs themselves, that is the drugs from the poppy plant, the opiates, and then all of the synthetic opiates that are used also in this talk, but we're really going to embellish on that in part two. So, again, I'm Dr. Dan Guerra. This is VerEvMed Studios, and again, it's March 12th, 2018. This is our company, VerEvMed. You can contact us directly at this web address and also in this email, and you can become a client of ours. And what we do is we go into the scientific literature, and we, first of all, look at the evidence that is of a topic that our clients are interested in. And then we look at that evidence, and then we verify it and vet it by looking at the preponderance of evidence in association with the foundation and coherence of that data and information relative to the foundations in science and the coherence of that science to even other disciplines. Okay, so that's what we do for our clients. We work with medical doctors, we work with veterinarians, we work with lay people, we work with lawyers, and all other walks of life. What we try to do is discuss with you what is going on in biomedicine at the research level, that is in the published research level. So let's get started with opioid receptors and pain. First of all, let's take a look at a paper that was published a few years back in the Annual Review of Medicine by Stein. And let's start talking about pain itself. Okay? Remember that analgesics, such as opioids, are used, of course, to blockade pain, right? So what about pain? Pain is divided broadly into two categories, so-called physiologic and pathologic. The physiologic, or also known as acute or nociceptive, is an essential early warning sign, which usually elicits a reflex withdrawal, such as when you feel a flame. And what it does basically is promote survival by protection from further injury. That type of pain is not the objective normally of therapeutic intervention, right? We're not trying to block that injury-inducing pain threshold so that you don't feel pain. You want to feel pain if something bad has happened so you can get away from it. It's not what drugs are used for. We're more interested in the pathologic, for example, neuropathic or chronic pain. And there, pain is an expression of maladaptation, or maybe just regular normal adaptation of the organism to tissue injuries. So in other words, this is pain after the injury or the insult has happened. Most painful conditions initially involve the activation of dorsal root ganglion neurons, or DRG neurons, I'm going to call them from now on, which give rise to A delta and C fiber innervating peripheral tissues. So those are known as nociceptors, and those tissues include things like skin, bone, joints, and viscera. That's where you feel a lot of your pain. Primary afferent neurons will transduce this noxious or pain stimuli into action potentials and then conduct them to the spinal cord. Transmission of the input from the nociceptor to the ascending spinal neurons and ultimately, of course, to the brain is then mediated by monosynaptic contacts or through interneurons. Endorphins, which are endogenous opiates or endogenous morphine, right, respond to pain and again blunt the pain. Okay, they don't actually shut it up, they just blunt it. All right, now let's take a look at endorphins. Endorphins come from a a polyprotein complex, for example, pre-pro-opiomelanocortin. Now notice this, POMC it's called. We'll have a signal peptide and we'll have various proteins embedded in it. Here's ACTH, okay, that is adrenocorticotropin releasing hormone. There's beta lipotropin. There's MSH, okay, that is melanocyte stimulating hormone. There's the gamma LPH, and there's beta endorphin itself. The compound is the endogenous opioid. Okay, and there's also a gamma MSH out here. So that's one polyprotein complex that is proteolytically processed to give you all of these hormones. Here's pre-pro-enkephalin, again with the signal peptide, and here's all the enkephalins. We'll explain to you what the structures of enkephalins are in a moment. And there's pre-pro-dynorphin, another in the endomorphine complexes, and there are various forms of that. And there's pre-pro-morphanin, FQ, which is a newer one on the scene. Okay, so let's take a look at what these structures look like. There's beta endorphin. That is your naturally occurring peptide hormone, which will modify the pain signal through the pathway we're going to discuss in a moment. There is metenkephalin. You notice that it is just a peptide fragment of beta endorphin. And there is leuenkephalin, which again is a peptide fragment from beta endorphin. Now notice how those are peptides. That means amino acids linked together via the peptide bond, right? They have very little, in fact, they have nothing biochemically in common with the plant alkaloid known as morphine. Morphine is an opiate. That's one of the most powerful opiates there are. It comes from the poppy plant. Notice its structure is nothing like the naturally occurring endorphins and enkephalins that we synthesize endogenously to blunt pain. Yet, we'll find out that morphine binds to the same series of receptors. There are actually 3 opioid receptors. Okay. Now, let's take a look at how this works. We'll get very, very general and then down into more accurate and specific. In a generalized pathway, the afferent neuron, which is here, of a sensory unit is the first in a chain of neurons that relays information ultimately to the central nervous system, which is ascending at the top of this slide. That includes the central nervous system, the spinal cord, and of course the brain. Now the afferent neuron is the first order neuron. It synapses with a second order neuron that synapses in turn with a third order neuron in the thalamus. The third order neuron then guides the impulse to the sensory cortex where it is perceived. Okay, and so you can see here we've discussed these axon, the entire neuron structure before in the last 2 talks, so I'm not going to go through that again. This is just a blow up of what it might look like with receptor cells in the periphery. These would be the sensory receptor cells, for example, in your skin. Axon branches moving up the axon, then the axon terminates again into the spinal cord and the CNS. Okay. All right, now here's more detail. Interneurons inhibit second-order neurons that transmit pains. So interneurons then are going to be where you're going to get endogenous opioids that are going to inhibit the second-order neuronal stimulus, which is the pain stimulus. Okay, so this is the ascending pathway to the thalamus. This is the descending pathway, right? Okay. So let's take a look at this. C fibers transmit pain signals from the periphery, and they inhibit those interneurons. However, the A beta fibers transmitting touch, for example, or pressure or vibration stimulate those inhibitory neurons. Hence, non-painful mechanical stimuli actually also decreases the pain threshold. The brain can also influence the perception of pain through the descending pathway, as shown here. That descending pathway coming from the periaqueductal gray, or the PAG, through the lateral reticular formation, through the raphe magnus, shown here, ultimately down into the afferent neuron, which is blown up here. Now look at this afferent neuron. You got the descending neuron coming with this flow, okay? You've got the interneuron here connecting up, right? Here's your afferent neuron over here, your ascending arch. And here's substance P being generated. Now, substance P is going to induce the pain response. But here you have the release of enkephalin coming from the second order neuron. So here you're getting this blockade of that pain signal. Okay, that's how the descending and the ascending complexes function together down here at the level of the original nociceptor. Okay, all right. So that's how the brain influences the perception of pain through that descending pathway. Now, neurons in the PAG up there, you see up here in the PAG, periaqueductal gray, that's part of the brain, communicate with the nucleus raphe magnus, that's shown there, okay, in the medulla, and lateral reticular formation. That's this general region right here, lateral reticular formation. And this is known as the spinothalamic tract. All right, now, neurons from these areas descend the spinal cord and synapse with the inhibitory neurons, which we're talking about here, which are going to be producing things like endorphin and enkephalin. And here we're showing the enkephalin response. This in turn synapses with the axon terminals of the afferent neuron, which is releasing substance P, which is inducing the pain signal. Okay. Cell bodies and dendrites of the secondary neurons to induce the inhibitory postsynaptic potentials. So you have a promotion of the pain response, for example, by the secretion of substance P, and then you start getting this pain blocking response from the descending system coming essentially from the periaqueductal gray matter area, and that is eventually down here at the interneuron area going to release the endogenous opioid. Here in this case, they're showing you enkephalin. All right. Now, just showing you another slide of the same thing. Okay. Here is the ascending nociception pathway from where the dorsal horn is. This would be in the spinal cord. So you get an injury. The injury then is transmitted along this afferent neuron. Substance P is generated. Okay, and you're sending that nociception, that pain signal, up to the brain, the tract we just looked at, the spinothalamic tract. Okay, now at the same time, endogenous opioids, for example, are stimulated, and we'll show you how, via a series of voltage-gated channels. And those endorphins then, or enkephalins, descend down this spinothalamic tract, okay, back to the spinal cord, where they again blockade the substance P effect of the initial pain stimulus. And that's why it says here opioids inhibit transmission, right? Now, if you wonder how aspirin works or ibuprofen works, it works right here at the very early stages for the release of substance P. Okay, that's not what opioids do. Opioids work after this is released, after the signal is received up the spinothalamic tract. Then they're sent to dope the system to decrease the pain threshold after you get the stimulus. Okay, it's a totally different thing. Other regions of this brain, okay, there's the periaqueductal gray, the LC is known as the locus coeruleus, and the NRM is the nucleus raphe magnus. We saw this already in the cutaway slide. So let's go through this. So basically, you have an inhibitory mechanism. That's how you make the pain threshold diminished. In injured tissue, there is a system involving leukocyte-derived opioid peptides. So when you have an inflammatory response, the leukocytes themselves will generate opioid peptides, such as endorphin and enkephalin. You also get peripheral nociceptal terminals carrying the opioid receptor, right? Because once you make the opioid, you have to have a receptor for it. In the spinal cord, inhibition is mediated by the release of endogenous opioids or by gamma-aminobutyric acid, that's the inhibitory neurotransmitter, okay, from interneurons, which activate then the presynaptic opioid and or GABA receptor on central nociceptor terminals to reduce the excitatory transmitter release, shown down here. The opening, I told you, is going to have to do with voltage-gated channels. The opening of postsynaptic potassium and or chloride channels by opioids or by GABA evokes a hyperpolarizing inhibitory potential in the dorsal horn neuron. During the ongoing nociceptive stimulation, then the spinal interneurons upregulate gene expression and they enhance the production of opioids. Right? The enhancement of those endogenous opioids then, again, buffer or blockade that pain response. That's what we mean by the descending inhibitory pathway. It includes the opioids, noradrenergic pathway, serotonergic pathway, and of course the GABA system itself. So a key region up here in the brain is the periaqueductal gray, right? And it relates to the rostral ventromedial medulla, which then projects along the dorsolateral funiculus to the spinal dorsal horn. There is your descending pathway. Okay? Good. Now, I told you that we were going to talk about opioid drugs, and we are. And so, especially in the second lecture that I'm going to do, so this is a two-part lecture. But what kind of compounds are we talking about? Well, here you've got some. Here's a fentanyl. Here's a strong analgesic potency, but has very little dependency potency. It's a selective mu-opioid receptor agonist. It's not usually used for management of chronic pain, and therefore tolerance and dependence aren't even relevant. Okay. There's buprenorphine, which is a very strong analgesic potency. It also has a tolerance. That means the more you take, the less it responds. You get a tolerance to the drug, and you also get a dependency, which is because of that tolerance. Now, notice what it is. Here we're talking about the receptor itself. It's called a partial mu-opioid receptor agonist and also a kappa receptor agonist. And they can precipitate withdrawal in individuals depending on other opioids that may be in the system. Only partially reversed by naloxone, and so naloxone is what's given as an opioid antagonist. Okay, there's codeine, relatively low analgesic potency, relatively low tolerance and dependence. Another really important one here, of course, is heroin, or diamorphine. It's basically acetylated morphine. It is a very potent mu receptor agonist. It's acetylated, as I said, as a prodrug, so it's more soluble and it's able to get at the site much more quickly. Very strong analgesic potency, the highest really, and very strong because of that tolerance and dependency possibility. Here's fentanyl. Fentanyl, strong analgesia, not so strong in tolerance and dependence, but still there. Hydromorphone down here, and then finally methadone down here. Okay, now that's a potent and selective mu receptor agonist, also a monoamine reuptake inhibitor. It works in multiple functions. Okay, we'll talk about methadone treatment later. All right, there's morphine, of course, the parent drug, very strong analgesic, very strong dependency, and you see the rest. There's oxycodone, very strong analgesic, pretty high in terms of dependency. Notice that the mu receptor is the one that's usually, there's three receptors, and the mu is the one for the opioids. It most commonly is the one that gives you trouble in terms of dependency and addiction. Okay, some few other of these synthetic opioids, which we won't be talking about further. So, what's some more that we can understand about the physiology of pain? First of all, you get a stimulus. For example, you get your finger pricked with a sharp object. You get a transduction. You get transmission up the spinal tract into the brain. Okay, ultimately through this spinal tract, through that up into the brain, you get to the raphe nucleus eventually, and to the periaqueductal gray, where the response occurs, and you get now the downward modulation. Once perception has occurred, the downward regulation, which includes things like the endorphins and GABA. Okay, that's the physiology of pain. There are 4 stages of it. So let's talk about transduction. That's the process of converting a painful stimuli, like a cut on your finger, to a neuronal action potential at the sensory level. So you have nociceptors, which are going to be there at the free nerve ending, and they transduce the painful stimuli into an action potential. You have chemical mediators which alter the membrane potential of that pain receptor. So those chemical mediators include potassium, proton, lactate, histamine, serotonin, bradykinins, and prostaglandins. Some of those are actually targets of NSAIDs, such as the prostaglandin pathway. As it says here, NSAIDs prevent the prostaglandin production by actually inhibiting cyclooxygenase. In one of my previous lectures, I talked about the cyclooxygenase pathway and the production of those oxy fatty acids, those oxygenated fatty acids. Now those generate pain. Okay, so that's what we're talking about here. They all basically come from the N-6 fatty acid, the omega-6 fatty acid, arachidonic acid, 20-carbon fatty acid with 4 double bonds, an omega-6 series. So that's okay. So now let's talk about transmission. The stimulated nociceptors transmit impulses to the CNS by means of specialized sensory fibers. What are they? We already discussed them. The A delta, which are large, they're myelinated, which means they have a coating of cerebroside and sphingomyelin. And they're involved in the transmission of sharp stinging and highly localized pain. Then you have the C fibers, which are smaller. They are unmyelinated, so transmission is slower. And they're involved in the dull, aching, and poorly localized pain that lingers, such as after you get some kind of injury, hours after that injury, you get that low, dull pain. Those are from the C fibers. Most sensory afferent pain fibers enter the spinal cord by way of what's known as the posterior nerve root. They travel to the substantia gelatinosa, and that's the area where pain can be enhanced or blocked. And there are cell bodies there for pain neurons located in that dorsal root ganglion, which we've told you about, DRG. So here again are the fibers, the A deltas and the C fibers. The important thing to remember here, the A deltas are myelinated. There's much less of that than there is of the C fiber. What kind of pain do they receive? Thermal, mechanical stimuli. Whereas the C fiber are more polymodal. They can include mechanical, thermal, and even chemical. Speed's very fast with the A delta fibers because they're myelinated, of course. The C fiber is much slower. Sensory quality of the pain mediated. You get the sharp stinging, cutting, or pinching from the A delta fibers, and you get that dull burning or aching from C fiber mediation. So more about transmission. Pain signals are transmitted by the afferent fibers. They enter the spinal cord through the dorsal horn, as I showed you previously. They synapse on interneurons. And then they cross the cord and project centrally to the anterolateral tract. The anterolateral tract has two divisions. The neospinothalamic tract, which I showed you, which carries the A delta fiber input, the myelinated input, and that projects directly to the thalamus and then onto the sensory cortex. And then the paleospinothalamic tract, which carries the C fiber input, that's the dull pain input, projects diffusely into the reticular formation, the mesencephalon, and of course the thalamus. So here it is again. You get the pain stimulus down here. They're calling it a noxious stimulus, okay? Chemical burn, or maybe just a high temperature, like you put your finger near a fire, or mechanical, such as something smashes into your finger. Here's all the peripheral activity down at the nociceptors. You get vasodilation. You get edema, which is swelling. Hyperalgesia means your pain response turns on. And you get a release of several chemicals, such as the prostaglandins. Then you're moving up through the pathway here. You get to the spinal cord. There's the dorsal horns we showed. There's the anterolateral tract moving through the brainstem and into the brain. Here's the thalamus. Those axons project to all the other areas of the brain. The limbic, which gives you an emotional reaction to the pain, of course. So like, ouch, that hurts. I don't like it. So a negative affect, right? Primary sensory cortex, the location. Where is the pain, right? Because that's going to involve the executive branch of the brain, the prefrontal cortex. And the cortical association is where the pain is interpreted. What kind of pain is it? Okay, so these are distributed. All these signals are distributed throughout the brain. So that then leads us to perception. That's when the brain actually interprets the pain as something painful, something that's hurting you. The result of neural processing of pain sensations in the brain is what we're talking about here. It's influenced by awareness. Oh, look what happened. Emotion. Oh, I don't like that. previous experiences, boy, that feels a lot like my hand is near the fire. And then expectations, culture, and physical status. Like all these things are built into how you respond via the prefrontal cortex and executive decision. There's also something known as the pain threshold. That's the level of pain stimulation required to be perceived. It's different for different people. We don't know much about why that's the case, but we do know that some people have much higher pain thresholds than others. So there is an executive decision there. Probably there's also talking to the limbic system to diminish the amount of emotion so that whatever pain's received causes less of an emotional response. So that leads to pain tolerance. That's the degree of pain an individual is willing to bear before seeking relief. And again, it varies among individuals. And then pain expression, way in which the pain experience is communicated to others, and that also varies, of course. See, when you're telling a doctor, oh, it's a sharp pain, or it's a blunt pain, or it's a burning sensation, right? So that leads to modulation. That's a very complex mechanism whereby synaptic transmission of the pain signal is actually altered. And of course, this is where it leads to our discussion of the endogenous opioids, enkephalins and endorphins. Okay? I showed you the descending pathway, and it originates in the raphe magnus, as I said, and it goes to the dorsal horn, and then it releases neurotransmitters that can inhibit the synaptic transmission of the pain signal. Opioids, including endorphins and morphine, okay, that would be an exogenous opioid, are mediators of that presynaptic inhibition, right? That's what's also known as modulation of the pain. The raphe magnus receives input from the periaqueductal gray, as I showed you, which is a high concentration of endogenous opioids, and the rostral pons, which secretes norepinephrine, which enhances this response. Okay, so opioid receptors here, finally we're getting to this. All right, the opioid receptors are expressed by central peripheral neurons and by neuroendocrine, such as pituitary, adrenal, immune, and ectodermal cells. There are three main types here, the mu, the delta, and the kappa. Mu being the most important, as we will see. Okay, so the opioid receptors, all of them, belong to the gamma subgroup of the 7 transmembrane G protein coupled receptors, or GPCRs, which have been the topic of several VerEvMed lectures. And they show anywhere between 50 to 70% homology amongst that gene family. There are lots of other pharmacologically determined subtypes that probably result from alternative splicing of the messenger RNA. So the mu receptor, systemic and peripheral is where it works. The delta, systemic and peripheral, the kappa, so they're all over the body. The effects, the mu receptor gives you euphoria. So this is going to be where morphine and heroin play a major role. Analgesia, which is pain killing. But it also gives you constipation and respiratory depression as it kills people. Peripheral only gives you analgesia, constipation, and reduced inflammation. Okay. And then there's the delta, gives you analgesia, convulsions, and angiolysis. Okay. Analgesia, constipation in the peripheral, and you can see the rest of it down here. Okay, the agonists, for example, in the mu receptor, morphine, fentanyl, endomorphines, and the beta endorphins, all of those endogenous are all binding to the mu receptor. At the periphery, you also get some analogs of those drugs. Okay, the antagonist, naloxone, for the systemic, and naloxone also works on the peripheral. Naloxone also works on the delta receptors, all the way through. All of the opioid receptors can be blocked or antagonized by naloxone. And there are other drugs also that will antagonize those various receptors. Okay. So perception of pain, as I said, starts when a painful stimulus activates the neural signals originating from the terminals of nociception. Those signals are propagated to second-order neurons in the spinal cord or brainstem and subsequently transmitted to specific higher-order brain regions that we saw in the thalamic tract. The pain stimuli ultimately activate ion channels, and those are present on the nociceptor terminals. Those are the potassium channels, the chloride channels, sodium channels. And they are going to actually be doing the transduction of the signal and they're going to depolarize the neuronal membrane. That's a major thing that happens. Okay, you're going to depolarize that, which was going to then alter the action potential, right? One such ion channel is the transient receptor V1 or the TRPV1, okay? Transient receptor potential V1 is one of the key ion channels. It's conveniently activated for purposes of research by the chili pepper substance known as capsaicin. That's the compound we actually have in chili pepper responsible for its hot taste. So why am I telling you this and showing you this drug, and I'm going to even go into more detail, is because when people want to study nociception, it's very convenient to use capsaicin as a model compound to induce a pain response and then look at this signal transduction cascade through the entire hypothalamic tract. The capsaicin is a very useful drug for that reason. What is capsaicin then? It's a plant alkaloid, neuropeptide substance P releasing selective agent from sensory nerve terminals. And there it is right there. Its actual structure is N4-hydroxy-3-methoxy-8-methyl-6-nonenamide. So it's 9-carbon fatty acid with nitrogen linked here to this phenylpropane chain. Now, before I became a biochemist, I actually was a phytochemist. When I did a master's degree, I was doing phytochemistry, and then I moved into biochemistry for my PhD, and I've been doing pretty much biochemistry for the last 35 years. But I really still have a warm place in my heart for the secondary product pathways that you see embellished in microorganisms, but in my case, the ones that I studied were in plants. Now, these are really beautiful pathways because they combine all the features of organic chemistry and molecular design in terms of how pathways converge in order to synthesize really complex structures. And there's no better one than when we look at something like capsaicin. So you have the phenylpropanoid pathway, which of course we don't have in animals, starting with phenylalanine, going through phenylalanine ammonia lyase, it's an enzyme I actually studied for my doctorate degree, making cinnamic acid, and then various steps later, coumaric and caffeic acid. These are phenolic compounds, ferulic acid, vanillin, and then vanillamide. That structure is going to condense with 8-methyl-6-nonenoic acid. It's 9-carbon fatty acid. You see it with one double bond. That actually comes from valine degradation, one of the ways it can, via the branched chain amino acid transferase reaction, making alpha-ketoisovalerate. Now, alpha-ketoisovaleric acid, interestingly, is also a precursor to pantothenic acid, which is the precursor to coenzyme A. Okay, so very interesting, very complex pathways. I love them. Okay. So anyways, that deaminated valine, you get alpha-ketoisovalerate. Now, you do actually make a thioester with coenzyme A, so not to confuse this with a substrate for synthesis. Now, you're just adding the resolved completed CoA molecule, making isobutyryl-CoA. Remember, you make a thioester to activate that carbonyl so that you can get into condensation reactions, basically, as shown here. So you're adding malonyl-CoA, which means you're using all the fatty acid synthesis machinery that I talked about before. You need fatty acid thioesterase to break one of those thioesters. You need acyl carrier protein, which I studied at great length in my earlier days, and the ketoacyl synthetase, KAS. You ultimately make the 8-methyl-nonenoic acid. You add dehydrogenate to put in that double bond you see there. And now you're able to condense using capsaicin synthase, and there's capsaicin. So 2 metabolic pathways converging to make this one compound, which, as we said, is going to bind to the opioid receptor, and it's going to cause the hot sensation you get from chili peppers. Amazing. All right. Now, let's get back to animals. The diagram in the middle is going to be described by both of the two sets of writing on either side, on the left and right. So let's first look at this. Now, you know that G proteins work by hydrolysis of trimeric G proteins. Trimeric G proteins have an alpha, beta, gamma subunit structure. So you have the G alpha and the G beta gamma subunits, which separate out. The former, the G alpha, inhibits adenylate cyclase and therefore cyclic AMP production, whereas the latter, the G protein beta gamma subunits, what do? They directly interact with different ion channels in the membrane. So you have two different systems going on simultaneously here. Okay? Sorry. All three opioid receptors can modulate pre- and postsynaptic calcium channels, suppress calcium influx, thus attenuate the excitability of the neurons and reduce the release of the pro-nociceptive substances. Okay? The opioid receptor activation leads to the opening of the G protein-coupled inwardly rectifying GERK channel, which we talked about when we were discussing ethanol intoxication and cannabinoid intoxication in previous talks, thereby preventing the neuronal excitation or propagation of the action potential. Opioids also inhibit sodium channels, transient receptor potential of vanilloids, which we just saw, the TRPV1, and even an acid-sensing ion channel called ASIC, as well as the excitatory postsynaptic currents evoked by glutamate receptors in the spinal cord itself. Ultimately, this results in decreased transmission of the nociceptive stimuli at all levels of the neuroaxis and profoundly reduces perception of the pain. So here we're looking at a leukocyte, which is leukocytes we're going to bind to their to a peptide to its receptor, okay? And when that happens, when CRF binds to its receptor, you get opioid peptides generated, and that's what these are here. So a leukocyte itself, in response to hormonal stimuli, will generate opioids, endogenous opioids. So leukocytes are stimulated by stress to secrete opioid peptides. For example, CRF, interleukin-1, and noradrenaline can elicit opioid release by activating each of their receptors. Okay? That's a corticotropin releasing factor. That's what CRF is. We know that this is interleukin-1, and we know that adrenergic receptors working through norepinephrine. All right. Now we also see here exogenous opioids binding down here. And see, here's where you're hooking up to the G protein complex, and you're diminishing that cyclic AMP. So you're working way down here. You're not, of course, generating them from the leukocytes, but this is where they bind to the same place on this afferent exon here. So there are the opioid receptors. They're binding to the opioid peptides that are generated from leukocytes. And there they are being generated from CRF or by interleukin, right, binding to its receptor, right, generating these opioids which bind to the opioid receptors, tanking cyclic AMP, opening up the calcium channel, right, endogenous opioids and drug opioids, right, the OPs, right, those are the you have endogenous opioids or endogenous opioid peptides, either one, the green triangles, right. Here you have the ascending arch, right. Here you have the generation of those receptors, right? You have the RNA being made so that you're able to generate more of the endogenous opioid, and that's all being turned on here in the dorsal root ganglia. So you make more of the receptor. The more of the receptor for CRF or IL-1 or NA, you generate more endogenous opioids. They bind to the receptor, and they are now what? They're inhibiting the pain response. Okay. They're synthesized in the dorsal root ganglia, as I said, those receptors, and they're transported along the intraaxonal microtubule to the terminals of the sensory neuron, okay, down here. Subsequent inhibition of the ion channels like the TRPV1 and calcium, and of the substance P release shown there. Okay, so substance P is being released down here, and when it's being released, you're going to start inhibiting that effect by the opioids. Okay? Hence, you have what's known as the anti-nociceptive effect. So that leads us now to a specific discussion of how the receptors are expressed, what induces them. We just looked at a general understanding that they are turned on. Now let's discuss some detail. There are alterations in the opioid receptor expression which occurs transcriptionally, translationally, and in fact epigenetically. Inflammation can induce differential regulation of those receptors. Altered upregulation is dependent on neuronal electrical activity and on cytokine release during inflammation, as we said. Axonal transport of those receptors is stimulated by cytokines and nerve growth factor, or NGF, and that's, of course, all going to be produced in inflamed tissue, such as epithelial tissue. The upregulation of opioid binding sites is due to an increase in the number of neurons expressing the receptors and the number of receptors per neuron. But the receptor affinity is not affected in this normal process. Okay, so we're going to stop now with this short initial lecture. We introduced you about how pain functions in the body. We talked to you about how the opioids, endogenous opioids, mute that pain signal from the afferent neurons up through the spinothalamic tract. We told you how opioids, endogenous opioids, are generated by the periaqueductal gray, and how you get emotional stimulation and executive decision on how to respond to the pain, and that the sending pathway is inhibitory, working through GABAergic pathways, blockading then those ion channels and slowing down the pain process by shutting off things like the production of substance P. So the next talk I'm going to do is going to discuss the opioid drugs themselves, the drugs that are being abused, and how that has caused the opioid crisis that we have here and worldwide, basically, but particularly in the United States is what we're referring to. So there's my Facebook page. You can get this VerEvMed lecture directly on there via the YouTube. You can contact me directly via my email address, or you can go to my Facebook. You could also contact us directly with this email address from the company, and there's our website. Please go there and you'll see what we do. And I really appreciate your attention. Now it's ready for me to do the initial sign off by saying bye for now, and look for part 2, which will be coming directly. Thank you.